and I will start from introducing our team members. First, Licia Celeri, whom some of you may know, recently moved to the UCSF, so that clearly facilitates our direct interactions and joint lab meetings, and also interactions with other spokes that are located in California. I also want to acknowledge Sarah Prescott, who recently moved for a postdoc to Harvard, but she was really a key person in my lab, who was this very brave graduate student who took on some of the initial aspects of the project and provided proofs of principle and generally a lot of data. Tomek, whom many of you met last year, but he couldn't be here this year, is doing a lot of our data analysis and communication with the hub. And I also want to introduce two new postdocs, very talented people who joined our team quite recently, Hannah Long, who joined my lab, and Ian Welsh, who is in Licia's lab at UCSF. And just to remind you, our aims, as we originally stated them, were to characterize the epigenetic landscape and transcriptomes of human and chimpanzee cranial crest cells, and to identify both conserved as well as species-specific cis-regulatory elements. And aim 2 was to analyze activity of this candidate human-specific craniofacial enhancers in vivo in mouse transgenesis. So what motivated this project and our thought that these data sets may in fact be useful? So I think a big challenge that the whole field is facing now is to understand how non-coding genomes, and in particular regulatory elements, encode human traits. From GWAS studies, we learned that a lot of the SNPs map to the non-coding regions of the genome. They're enriched within regulatory elements such as enhancers, and both for the human variation and human complex disease, a lot of the genetic explanation seems to be within the regulatory parts of our genome. However, our understanding of how changes in regulatory elements such as enhancers reveal themselves through developmental process to give rise to complex phenotypes is still fairly rudimentary. And we thought that the time is ripe to start addressing these questions due to multiple developments both in the field and in our labs in particular. And one is the ability to systematically identify cell type specific regulatory elements using epigenomic mapping strategies. But also our ability to model aspects of human development in the dish using pluripotent stem cell based models. And finally, through the follow-up analysis in model organisms and also in vitro, we were able to convince ourselves that even though clearly in vitro models have their limitations, they are nonetheless an extremely useful tool for understanding human phenotypes. So we also decided to think about the evolutionary aspect of so in addition to creating a lot of human data sets, we wanted to add evo-devo aspect to our study. And this was really motivated by elegant work from other labs like Sean Carroll's or David Kingsley and others, showing that studying evolution of closely related species can be indeed an extremely powerful tool for uncovering genotype-phenotype connections. And indeed, these labs have demonstrated that change in enhancer sequence can drive change in the phenotype, such as wing patterning in Drosophila or skeletal changes in stickleback fish. And we wanted to extend this type of evo-devo approach to higher primates in the strategy that we refer to as cellular anthropology. In other words, using higher primary cellular models to study both enhancer landscape evolution, with the but also enhancers that are active both in the context of humans and other primates, with an ultimate goal of understanding the genotype and phenotype connection. And I hope I can convince you that this evolutionary lens can really reveal information that is relevant not only for people interested in evolution, but also for understanding fundamentals of human development and disease. This group does not need introduction to the neural crest, and all of you know that most of our face and head is derived from the cranial neural crest during development. And indeed, this is therefore not surprising that cranial neural crest-derived structures are thought to have played a key role in human evolution by facilitating not only uniquely human facial appearance, but also a number of adaptations associated with upright posture, speech articulation, and large brain size, etc. But as you also appreciate, the cranial neural crests from humans and higher primates are essentially inaccessible in an embryonic context. They arise at 3 to 6 weeks of gestation. This is transient migratory population. So we had to learn how to derive them in vitro from pluripotent stem cells, ES cells or iPS cells. And I mentioned this in the previous meeting that my lab has developed methods to obtain neural crest cells from these pluripotent stem cells that have both the molecular characteristics of the cranial crest that's seen in all organisms, but also cellular characteristics. Namely, we can take these cells and transplant them to the chick embryo, and they will migrate, assuming correct positional identities. And they also retain this broad differentiation potential characteristic of the human crest. And Sarah, when she was a graduate student in the lab, extended this to other primates, namely the chimpanzee, and also we're starting to work with gorilla as well. And this slide is summarizing a lot of her work, which essentially a key advancement here was to ensure that not only that the chimp cells that we're deriving are truly neural crest cells, but also that we can minimize heterogeneity and stage the cells correctly for comparisons with human cells. Otherwise, developmental differences were dominating the analysis over species differences. So in the end, we used 6 different surface markers and had to go quite in depth to allow for comparisons between those two populations. But nonetheless, this brings us to the data sets we're generating for the FaceBase, which are from neural crest-derived cells from several human and chimpanzee individuals. And those include ChIP-seq data sets for some neural crest transcription factors such as AP2 alpha, germinal coactivators like P300, analysis of the hypersensitivity patterns with new technique called attack-seq, and also ChIP-seq analysis of chromatin landscapes, including modifications like K4 monomethylation, K27 acetylation, K4 trimethylation, and not mentioned here, also K27 trimethylation associated with polygam silencing. And we're also generating RNA-seq data sets to allow for comparison of the changes between the epigenome and transcriptome. And based on the combinatorial chromatin signatures derived from that, we can systematically annotate enhancers that are active in human neural crest cells and distinguish these elements from other regulatory elements such as active and poised promoters, etc. So this slide just shows you that our epigenomically mapped enhancers can indeed, even though they were derived from this in vitro system, drive craniofacial gene expression in vivo. And in this one example, we cross-reference our human active enhancers, active in our in vitro model, with the Vista enhancer browser, which is an effort led by Len Penacchio and Axel Wiesel, to test activity of different regulatory elements, human regulatory elements. And we detected almost 250 regions overlapping. And as you know, VISTA does not have any craniofacial bias. There's analysis of enhancers from different organ systems. And nevertheless, when we looked at the enrichments observed for our neural crest enhancer elements in terms of the patterns observed in VISTA, we saw high enrichment for activity in branchial arch and facial mesenchyme, consistent with the idea that we're indeed mapping craniofacial enhancers using our in vitro system. And this also illustrates really usefulness of resources such as VISTA, and hopefully this will extend to the FaceBase as well. Okay, this is not showing properly, but nevertheless, of course, we also wanted to know whether we can use this epigenomic strategy to map regulatory divergence between human and chimp neural crest cells. And the idea here was simple, which is that most of the epigenomic patterns should be invariant, since we're looking at the same cell type between two closely related species, but they will also be able to identify differences which can come either through structural variation between the genomes or through quantitative change at orthologous regions. So indeed, we've done analysis of or comparisons of epigenomic patterns from multiple human and chimp individuals, and indeed most of the epigenomic patterns are highly similar between human and chimp, as you would expect, but we also see biased enhancers or genomic regions that have enhancer signature that is strongly biased either towards chimp, as shown on the left, or towards the human, as shown on the right. And then we can systematically quantitate these changes and compare either different individuals of the same species, so different humans, and that's highlighted in red, or human and chimp, and this is shown in blue. So you clearly see that variation between the species is higher than variation within the species, and those and allows us to systematically genome-wide identify either human-biased or chimp-biased regulatory elements. So I would like to spend some time discussing, beyond generating these data sets and uploading them, I would like to spend some time discussing what we can actually learn from this type of data and how can it be useful for the community. So in data that don't have time to show you, we demonstrated that in fact these epigenomic biases in signatures are associated with differential ability of these enhancers to drive gene expression in assays like luciferase, reporters, or star-seq assay. And also what these assays demonstrated that the activity, that the epigenomic bias is driven by differences in an enhancer sequence between human and chimp. But interestingly, these differences are small, so we typically see 3 to 6 nucleotide changes between human and chimp that is sufficient to drive differential enhancer activity. So this gives us an idea that because we're working with genomes of such high orthology, we can use interspecies genetic diversity essentially like a large-scale enhancer mutagenesis screen. So what we have done, we scanned for all different transcription factor motifs enriched at crest enhancers, and we developed a quantitative measure of a difference of the fit to the consensus between human and chimp sequence. And we also developed quantitative measures of differences in epigenomic signatures. And then we correlated those two variables and derived correlation coefficients. And what you're looking at in this big matrix is essentially different motifs, so different positional weight matrices for different transcription factors versus different epigenomic features. So what have we got from this type of analysis? So first, we got a class of motifs that were positively correlated with gain of active signatures. In other words, gain of the motifs correlates with gain of enhancer activity. And not surprisingly, these motifs are in some of your favorite neural crest transcription factors, but also predict many novel neural crest regulators. We also got a whole class of motifs that are negatively regulated, meaning that loss of the motif correlates with a gain of enhancer activity. So this suggests that there's actually substantial input of repressive activities into fine-tuning of neural crest enhancer activity in the context of both evolution and development. And we're looking at some of those predicted repressors and their potential involvement in fine-tuning regulatory functions. But we also got one really big surprise from this data. So when you do this type of analysis, you rarely expect to find a singular outlier that is extremely predictive of the changes in your features. But this is indeed what we got. We got a one single long motif, a novel motif, which we termed coordinator, which was extremely predictive of any epigenomic feature that we looked at. In fact, could alone explain about 40% of the epigenomic variation. And not only that, coordinator is also predictive of binding of other neural crest transcription factors at enhancers, even though those transcription factors may have unrelated motifs. And it's enriched at crest-specific over pleiotropic enhancers, as we determine by comparing with 50 cell types from the ENCODE data sets. And which is an unusual feature by itself, is sufficient to drive expression in reporter assays in neural crest cells. So this really got us an idea that coordinator perhaps functions in a similar way as it has been reported previously in early Drosophila development for a very famous pioneer factor, Zelda, which binds to the so-called tactile motif. And tactile motif looks nothing like the coordinator, but we think there may be a conceptual parallel because Zelda licenses enhancers for activation. And through promoting active chromatin states, facilitates binding of other transcription factors. So we think that by studying evolution, we might have uncovered a similar mechanism operating in the context of the neural crest cells, and we're now purifying a regulatory complex binding to the coordinator. Now, by identifying a class of the functional mutations, so the changes that actually functionally matter, we can now go back and compare across primate genomes and do reconstructions of the ancestral state, and then try to see whether change in the coordinator motif represented human-specific gain, chimp loss, human loss, or chimp gain in terms of the evolutionary history of an enhancer. And interestingly, we saw that in our human biased enhancer, we saw roughly equal contribution of chimp losses and human gains, but really very rarely any human losses or chimp gains. And conversely, in the chimp biased enhancer, we see human losses or chimp gains, but never chimp losses of human gains. So this really shows you that both loss and gain are likely to contribute to the change in regulatory activity in evolutionary system. And we were also very compelled to go back to other genomes like Denisovan and Neanderthals, because now we have actually really decent quality genomes for those species, and ask about evolutionary history of these coordinator motif changes. And as you may expect, most of these changes happened prior to the split of humans from other hominins, because we're much more genetically closer to Neanderthal than we're to the chimp. But nonetheless, we identified a class of changes, class of mutations that appear to be unique to humans and not present in either Neanderthals or Denisovans. And I think this is quite exciting because we actually have a rough idea how Neanderthal neural crest derivatives look like, because there are skulls. And I'm really hoping that I could initiate a discussion with people like Benedict Hallgrimsson, who probably thought way more about the Neanderthal morphology than I ever did. Can we eventually make a link between this limited number of genetic changes and the morphological changes that are seen in Neanderthal skulls as compared to humans? Okay, let me get into good stuff. So it's really, of course, important to validate our data in vivo. So here I'm showing you an example of a human biased enhancer, which has two human-specific changes in coordinator motif. And as part of the FaceBase project, we're working closely with Richard Selleri's lab to clone some of those orthologous sequences and analyze their activity in the context of the mouse. So here, as you can see, that in this example, human sequence has gain of regulatory activity. So mouse sequence, chimp sequence is still active in olfactory placode, but there's a gain of multiple expression domains for the human ortholog, which is consistent with a gain of epigenomic signature in our crest cells. And here's another example where enhancer is actually really lights up a lot of the neural crest derived structures, and it really lights up the face as well. And corresponding chimp sequence is actually fairly negative. So we're going ahead with testing more of those in the context in vivo, but so far data looks really great for all the regions that have been tested. Of course, this wouldn't mean much unless we can link it to the changes in gene expression. So through our RNA-seq data, we see that species bias enhancers are indeed associated with genes showing species bias expression in the neural crest. And it's a lot of interesting genes that are on the list. And when we do ontology annotation of the species bias enhancers, all the top categories pop up are craniofacial. And indeed, they are associated with really quite interesting craniofacial processes. And here I'm just showing you a couple of examples of genes that are really interested and show species bias in an expression along with species bias enhancer patterns. So on the chimp bias side, a couple of interesting examples include PAX3 and endothelin receptor A. And on human bias side, I think one of the most compelling examples is BMP4. And BMP4 has already been studied in the context of Darwin's finches, and it's been associated with changes in morphology, in beak morphology in Darwin's finches. And it's interestingly enough, in our data set, BMP4 is human bias in expression and epigenomic patterns, and elevated expression of BMP4 in the mouse, that was a study published previously by others, specifically in the neural crest and the Wnt1 Cree driver, results in jaw size reduction and rounding of the skull shape. And in my naive approach, at least these are the morphological changes that are somewhat reminiscent of the ones that occur in human morphology as compared to chimp and other organisms. So this is just meant to illustrate that a lot of very interesting genes are on our list, and we're hoping that our data sets could be used to as a follow-up resource for morphological studies. And regarding that, I also want to mention that species bias enhancers are not distributed evenly across chromosomes. So when we, again, develop quantitative measure and the sliding window of enhancer bias across the chromosomes, there are regions that really seem to be particularly enriched for bias. And this is not because they have more enhancers. We controlled for that this does not simply reflect enhancer density. And if you look underneath, those regions contain clusters of bias enhancers, so not single enhancers, but whole clusters are biased. And you can identify those clusters of what we call them high regulatory divergence across the whole genome. And the interesting part about it is that out of the 5 genes that have emerged in GWAS studies as associated with normal range facial variation, 3 are actually on our list of clusters of high regulatory divergence. So I think what this suggests that perhaps similar mechanisms that drive variation between us and the chimps can also contribute to the intraspecies variation within humans, and perhaps some of these clusters of regulatory divergence deserve a closer look in morphometric and genetic studies of human facial variation, which are done by some labs involved in the FaceBase. So I'll skip summary, and I'll just want to discuss, as the last slide, FaceBase deliverables. So we, as I already mentioned, we generated most of the genomic data already. We have ChIP-seq data sets for histone modifications, P300, transcription factors, ATAC-seq data, and RNA-seq data. Those, most of them are actually already in the pipeline, waiting to be published on the website. We already submitted them. Those were done from 3 different genetically unrelated human lines, 2 different chimp lines. Many have, even within the same line, have been done in replicates to increase robustness of the data, but reproducibility is generally quite excellent. And we've already tested 5 constructs for looking at the enhancer activity and enhancer activity bias in the mouse. So those look very good. I also want to mention that we initiated collaboration with Axel Spoke to be able to test more of these regions, and also initiate other collaborations and integration of our data sets and their data sets. I think Axel will mention it in his talk. Yang Chai already mentioned a collaboration that we're initiating as well. And we also think that it would be exciting to introduce at some point functional manipulation with genome editing to not only describe the activity of enhancers, but also address their impact on morphology. Thank you, and I'm out of time.
Okay, we got a question over here. Hold on.
Joanna, this is, okay. Joanna, this is absolutely beautiful work. Question about how much intra-species variation do you have? So, for instance, when you look at your 3 human lines, do you actually find specific areas that have variation? And then if you look at, larger studies in the population, you maybe haven't done this yet, but you probably have thought about it. Do you think that those are areas that would have variability? Do you think that those are more the areas rather than the operator? I mean, what do you think is going to be the variation, the source of variation?
Yes, so we definitely see variation between the humans. For the chimp-human comparisons, we focused, we essentially demanded that there is a substantial variation between, so there's essentially activity in all humans and high activity in all humans as compared to chimp, or vice versa. So to really focus on so-called, more fixed activity. But even within those, with highly quantitative ways to measure bias in signatures, which we have now, you can still detect, to lesser degree than with a chimp, you can still detect intraspecies variation. So we definitely think that even the same enhancers could be that are different between us and chimp could be the source of variation, and then there are additional ones that we didn't count in the analysis because they had too much variation.
Let's see. One more question.
Joanna, so you've shown that they're cranial neural crest derivatives by putting them into chicken. Do you have an idea of more precisely where they might map in terms of time and position? In terms of
no, we haven't done. So we only know that we can put, they will migrate to the, and in fact, depending on where on the AP axis you put them, they will migrate either to frontal nasal prominence or to the mandible, and they will definitely, reside there. We haven't pushed the experiment much further, but they can definitely, their final destination seems to be clearly imprinted by the environment they will migrate in, and they will go depending on where their migration originated from.